Hello, everyone, and welcome. I'm giving everyone a few seconds to join from the waiting room. I see people are still joining. Thank you so much. Always great to see familiar names on the list, as well as new names. And again, we are committed to providing these updates every week, and it's really great for us to be here with all of you. So this is our regular weekly platform trial webinar, and I'm Sabrina Paganoni. I'm speaking from the HEALEY Center for ALS, and today I'm here with Dr. Cudkowicz, as well as our patient navigator, Catherine. So if we can go to the next slide. As for those of you who are new, as you know, the goal of the platform trial is to screen new drugs rapidly and efficiently. We want to get answers and determine the next steps. So far, we have included up to 7 drugs in the platform trial, and the first 4 had completed enrollment and reported results. As you know, the first 2 had negative results, but the next 2, regimen C and D, had positive results, so much so that they're moving on to later stage trials, and we can talk about it in the Q&A portion as well. Now, in terms of what's going on right now, in terms of participants who are participating or can participate in the trial, we have 1 regimen that's just completed enrollment. That's regimen E. We just completed enrollment. That means that we included all the all the people that we wanted to include in terms of our max sample size in the regimen, and now we're following them. We're following them as they receive active drug. That means that now we know kind of our overall timelines and when again when we expect the randomized control portion of the trial to end, because we have again we have enrolled everyone, but we need to follow them before we can obviously even begin to analyze the results. Now, in terms of the next 2 drugs, regimens F and G, they are in startup. I would say that we're very close to kind of getting started. That's going to happen, over the next few weeks. That's, always a busy time when we are in that startup phase. There's lots of moving parts and vendors and materials that need to be secured so that, we can have everything organized at all sites. But we're certainly making great progress. So we will be enrolling new participants soon. I told you earlier that regimen E completed enrollment, but we're going to accept new participants very soon for regimens F and G. Next slide. And as a reminder, for those of you who are new, the schema is the same for every regimen. Participants are randomized 3-to-1 in favor of active to placebo. So we have many more people on active than on placebo, but then placebo data is shared across regimens, and that's really what gives us that statistical power and efficiencies, such as fewer people on placebo. So it's really great that we can work collaboratively with so many industry and industry partners to make this happen. And then after the placebo control portion of the trial, which lasts about 6 months, then participants can enter what we call an active treatment extension. So they'll be able to receive active drug regardless of their previous assignment to active or placebo for for an extended period of time, so that we can basically compare the data from the people who started on active, so the early starters, to the people who had a delayed start. And this is to say that basically this trial is one trial, both the randomized period and the active treatment extension, and we get very valuable data from both phases, from both periods. And again, all of this contributes to our understanding of the drug. And right now, again, we have many participants that are at different stages here. We have for regimen D, the one that just had results. We still have participants in the open label extension. For regimen E, the one that just completed the enrollment, we have people both in the randomized period and active treatment extension. And for regimens F and G, we're about to start enrollment. So this is to say that this infrastructure is always active, lots of things going on at all times, because participants are in different periods, again, depending on the specific regimen. And we want to make this as quickly and as efficient as possible so that we can continue to have results. Next slide. So in addition to testing drugs, as you know, we are learning a lot about the science, about ALS. And so every participant, whether they are randomized to active or placebo, whether they are in one regimen or another regimen, they all contribute incredibly important data related to biomarkers, whether it's genetic information by giving a sample, donating a sample for whole genome sequencing, or other biofluid biomarkers. We're measuring neurofilaments, we're measuring, we're collecting spinal fluid, which is so important to test some of the new biomarkers that have just been developed for ALS related to some very specific mechanisms. And then these, we think, these advances in science really can really make us better equipped to really tell if drugs work and to learn more about ALS. And so that's why we're collecting all of these materials. In particular, spinal fluid is particularly important to be able to measure the newly discovered biomarkers that have just been shared and published. Also, as you heard, from the press release, we are really gaining great knowledge from speech analysis and new emerging digital biomarkers. So again, all of this contributes not only to testing the drugs, but also to ALS in general. For new regimens, we're actually going to have new outcomes. Many of you have asked us about ROTS, which is a patient-reported outcome that will be included in upcoming regimens. So we continue to learn about ALS and contribute to the science by sharing our results. Next slide. And this is kind of a snapshot of the enrollment numbers. Again, thank you so much to all of you who have participated, who are supporting people who are participating. Really, we couldn't be doing this without you. Since we started, we consented over 1,000 people, which is amazing. Over 500 people have enrolled in the open label extension receiving active drug. And so far, again, the participants have been distributed across 5 different regimens, but then the placebo data is shared. That's why we're learning more together. Next slide. We are starting to enroll for the new regimen. Again, we are accepting new participants very soon. And in particular, right now, we have 5 sites that are active for the new regimen, regimen F, which will test a drug made by Calico in collaboration with AbbVie. So those are the sites. As always, for those of you who have been following us from the beginning, we always share names of sites so that you can contact sites that are close to you if you're interested in participating. And all the links and QR codes on the slides will bring you to our website. We keep posted and updated so that you can see who to contact. So if you can go to the next slide. So for example, if you want to check the online status, the status of your site online, you can go to the link that's displayed here, and you can see whether they are enrolling for F or not. I do want to point out that the other sites that are listed as active, not recruiting, they are very much active. They're still active because they're participating in the previous regimen. So for example, they're still working with participants, for example, in the open label extension of D, as in David, or in the randomized portion of E, or the open label extension of E as well. So they're very much active. They're just not ready to recruit for F, the new drug, yet, but some are recruiting for F as well. So that's really great news. Next slide. And we have new resources on the MGH website. I do want to give a shout out to Catherine and other people on our team that really put together brochures to learn more about regimen F. One of the key features of regimen F is also that we're measuring innovative, brand new, really cutting edge biomarkers in the spinal fluid. So we have a lot of information about what it means to donate spinal fluid. That's incredibly important for us, and specifically for this regimen, because again, we have new science that really tells us that by measuring these newly published biomarkers, we can really learn more about ALS and about this mechanism of action. So again, if you have any questions, please go on our website. You can download the brochure. Everything can be viewed in digital form or paper form. And as always, Catherine is available to answer your questions, or you can ask us questions on the website, on the webinar. Next slide. And I do want to mention that, again, we continue to learn from our trial. This is sort of a continuously active trial on many fronts. And so we are going to have a community webinar open to the public on Monday about our recent regimen D updates. We got our first results for regimen D testing predopidine. We had a press release. You can view the press release on our website, but most importantly, we welcome your participation on the community webinar on Monday. And so that's our next appointment for results. And then to the next slide, I have another slide here. This is, again, our contact information for those of you who are new. Our patient navigators, Catherine and Alison, can always send you these information links or answer questions. And with that, I think we can stop sharing the slides. And I think Dr. Cudkowicz, I don't know if you want to start taking questions.
Yeah,
or
sure. Yeah. Thank you. Welcome, everybody. Please put in your questions. We have one already, which is about the people who get placebo during the randomized part of the trial. When did they find out what they were assigned to? And this person knows that all the trial participants are in active treatment extension phase when they're what we used to call open label extension.
Yeah, no, that's a great question. So as I alluded to earlier, the active treatment extension phase is very much an integral part of the trial, because we are basically learning a lot from that part. And because of that, we cannot reveal the previous assignment to active or placebo until the study is ongoing. So if you are a person who started earlier, obviously it means that, you know, we have to wait until the entire study is coming to basically to completion to be able to find out assignments. So that's because, again, there are statistical reasons and regulatory reasons to do that, so that we can maintain the scientific kind of integrity of the study. And I don't know, Merit, if you want to add to this.
Yeah, no, I think you got it completely right. We're actually required really to keep the blind throughout the active extension period of what people are on the double blind, so that when we're ready to analyze that, we can analyze that with the highest, you know, data integrity.
Yeah.
And that was a very successful approach, obviously, for Amylyx and the approval. So so once that part is done, then we'll absolutely be letting everybody know what they were on. So there's a question about how quickly do we anticipate adding sites for regimen F?
Yeah, so we are really focusing a lot on adding sites for regimen F. So we are working closely with the sites, and again, with there's some training that needs to happen, and also there's a number of things that need to get to the sites. So, for example, the actual drug or the lab kits. I will say that, you know, we're still waiting for some of the lab kits from our vendor that, you know, that could get to the sites. That, you know, again, some things are not under our control, but we're pushing as much as possible, again, to have all the supplies so that the sites can start enrolling.
I think of our 70 sites, we have 30 of them already who are through the ethics review, and which is really great. So the three things that are really important are getting that ethics review of the consent form, the contract with the sites, and then the training. And so I think I would hope that in March we'll have more than 30 sites open, and then the rest kind of April. So in the future, do you predict ALS treatment being a mix of drugs, or will that require new trials because of the interaction between drugs?
That's a great question. I mean, that's a good problem to have, in a sense, because, you know, the reason we didn't have to worry about interactions until very, very recently in ALS is because there was basically only one drug, you know, riluzole, since the 90s. And then, you know, obviously edaravone came more recently, and even more recently Relyvrio. Now, I think, you know, right now we are able to add drugs to standard of care, meaning that people can take 1, 2, or 3 of the previously approved drugs. But it is possible that, you know, for new trials, depending on the drug-drug interactions and the safety profile of new drugs that are tested, you know, there could be drugs that interact with each other. Right now, again, we haven't had that problem, and we think that all these drugs can be additive, which is the good news. In the future, as we develop more, at some point, we may have to prioritize or really try to figure out which subgroups responds most to specific drugs. So try to personalize the treatment a little bit more. But that will also be good news, again, as we develop more treatments.
The next question I can answer maybe it's whether we have data on neurofilaments in blood and do they relate to response to drug. So we have all the data now from regimen A through D at baseline and then over time. And we just got those because we had to run them all at one time because of the assay. It has variability if you do it, a 20 here and 20 there. So of the ones that we've gotten, we've fully analyzed regimen D, and now we're working on regimen A through C. So I don't have the results for A through C yet. But for D, we showed that neurofilament went down. The most it went down was 40% in the group that was clinically progressing faster and had the largest clinical response. Now, the numbers that were a little small, and we'll show that again on Monday in our webinar, but it definitely was, in the right direction and thematic. And hopefully soon, in the next couple of weeks, I think we'll have analysis for A, B, and C for neurofilament as well. But since we're talking about blood, I have to say, after spending the last 2 days at the Packard meeting, which is a heavy science meeting down at Hopkins, all the exciting biomarkers, like Sabrina said, are in spinal fluid, the ones that really measure the biology of what's going on in the cells with the TDP-43 protein. So I do worry that blood is not, at this point, going to be our best biomarkers, that we really do have to look at spinal fluid. And so in the new regimens, regimen F and G, we're really going to try to have ask people to donate their spinal fluid twice, once at the beginning and once at the end of the study, so that we can really tell if the drug is hitting the biology of the illness. And so we're going to spend some of these sessions a little bit about spinal taps. We have a video and we have some information about it. And we're also asked some people who have had them. There are many patients who have had them, especially people in some of the gene therapy trials where they have them every month, to kind of just say what it's like. And but I think that's going to be really important. So the next question is, since stopping progression of ALS will likely require cocktail drugs targeting a variety of pathways, why not have a trial where some trial participants are testing many drugs at once? This might make it harder to know which drug is most responsible for success, but given the stakes, isn't this something that should be considered in the future?
Yeah, absolutely. It's a great question. And I think it's something that absolutely should be considered. So right now, in a way, we are already doing it, kind of a segue from the previous question. So we're already allowing participants to take 3 drugs that are approved for ALS. And on top of that, we add an investigational product. So right now, all these drugs actually have had different mechanisms of action. So the drugs that we're testing have different mechanisms of action compared to the ones that are FDA approved. So we hope, we hypothesize that they will lead to additive effects. Now, I think that in the future, again, as more drugs are available, people may have to prioritize, or based on their own other drugs that they take at home. Again, that could be a more personalized package. I don't exclude the fact that we could have like an actual cocktail of drugs, let's say 3 or 4 drugs at the same time. It will depend on the mechanism of action. I know some colleagues are actually trying to develop some of these combinations, and I think that would be very, very interesting. Well, if you think about it, Relivrio is a combination of 2 drugs, so clearly it can be done. It just needs to have a scientific rationale.
Absolutely. So there's a question, what are the biomarkers from spinal fluid versus blood?
Yeah, so I think you already were starting to answer that with the previous comments. So that, for example, in blood, now we know that we can measure neurofilaments very reliably in serum or plasma, and those measurements are sort of, reflective also of what happens in the brain. So we don't need to, for neurofilament, to go necessarily and get the spinal fluid to measure that. Now, for other more recent biomarkers, that those are the ones that Dr. Cudkowicz was mentioning and were recently published and presented at the Packard meeting as well. They are really looking at very specific mechanisms or very specific proteins and mechanisms that happen centrally related to some of the very causative pathways in ALS. For example, many of you have asked, or in the past we spoke about TDP-43, which is a protein that accumulates in an abnormal way, in a different way, and becomes sort of toxic, a toxic accumulation in the cells of people with the majority of ALS. And so our colleagues have devised way to actually measure exactly some steps of that pathway, but those assays are only available in spinal fluid. That's why they're so important for some of the upcoming biomarkers.
We invited Jeff Rossi and Nick Maragakis to one of our Thursday meetings to tell us about all those new biomarkers in the spinal fluid. So that should be coming up soon. Just waiting to see if there's more questions.
I think there were a couple of questions that were posed previously. I know that Catherine also keeps track of questions that are sent to her by email. And so one question is, what are the steps to get involved in the platform trial and where to start? So I was wondering, Catherine, I know you've been receiving this question, I guess for people who are listening and don't know about a site or who to contact, what's the first step?
Yeah, so this has been one of the frequent questions in my inbox this week as we get started for Regimen F. And it kind of depends on where you're at. So if you are seen for your ALS care at a clinic that also offers a platform trial, that can be a pretty easy foot in the door to just kind of broach the conversation with your clinical team, and then they can connect you with the research team. If you don't go to an ALS clinic that's offering research, you can always reach out to me. I put my email in the chat. And then we can talk about local site options, or you can look at the site map to see. I always recommend doing that after looking at the eligibility criteria. So if it seems like you could qualify for the trial, then really the first step to getting involved is reaching out to a study team that's nearby or would be feasible to get to.
Thank you. So another question that came in before was how many in-person visits are required for Regimen F, and how does it compare to Regimen E?
So it definitely, there's fewer in-person in the sense that Regimen E had some extra infusions. For the Regimen E, because it was an intravenous drug, people had to get the first 4 infusions at the site and the rest could be done at home. So that's why for they had more. For F and G, they are oral drugs, meaning you can take them by mouth. And so we only have, I think, there's a couple of screening visits, and then I believe it's 6 or 7 visits to the site for the randomized portion.
Then there's a question of whether Tatka is still exclusionary, and if so, why?
Yeah, so Tatka is still actually in an active clinical trial, in a phase 3 clinical trial. So Relyvrio does include Tatka in combination with phenylbutyrate, and other investigators are trying to figure out if Tatka itself has an effect on ALS. And so in order not to confound the findings of this study, and because Tatka again is still considered an investigational treatment and is being tested in a different trial, it's exclusionary like other investigational treatments. I would also say that the Tatka that's been tested in the trial, and that's also in Relyvrio, it's pharmaceutical grade, as opposed to the Tatka that's generally obtained online. That it's a little bit unclear again what the effect of that would be. So again, to avoid confounding, unfortunately, that's still exclusionary.
Thanks. There's a kind of related question about a different drug. And can people take methylcobalamin B12 while in the trial?
So for the same reason, the ultra high dose, which is a very high dose of B12 in the form of methylcobalamin, that's also exclusionary, again, because it's still an investigational or experimental product. Now, if somebody takes lower doses of B12, those are allowed because those are not sort of the trial doses.
Someone mentioned that when they were first diagnosed, they had high creatinine levels, and I think that might be CPK in their blood. Is that part of a regular blood test?
So yes, the CPK, I generally obtain that as part of regular blood tests. Everyone has their own different labs that are ordered for diagnosis, but I would say that's a reasonable lab to monitor. And then basically it gives us an idea of sort of the muscle breakdown that can occur as a consequence of motor neuron problems.
Also asking whether we're monitoring T regulatory cells.
So not in this particular study. So we have done that in other studies where TRegs are actually infused or other molecules that modulate TRegs, which is an immune cell. So if for those trials where we were specifically targeting immune cell function or Treg function, yes, we could measure Tregs. I would say that there's many different ways of analyzing Tregs. They're not standard tests that you can just sort of, send to a regular lab. They're actually very time. So to monitor really number and especially function of Tregs, trying to see if they're activated or the status, it's actually a very time-consuming test, and it has to be done fresh by specialized staff. So it's not something that can be easily done. And in this particular, for now, we haven't had drugs in the trial that specifically target that mechanism. But in the future, obviously, that could change.
Yeah. As we're getting to the last couple of minutes, I want to first just remind people about the webinar on Monday. This is the open webinar to share the results from the 4th regimen, Regimen D. We have shared it with the participants as well as the sites and obviously in the press release, but we wanted to have a chance to show the actual slides and data to all of you and the broader ALS community. As we look back at the first 4 drugs, in 2 years and the time usually it would take to get the results of 1 drug, we got the results of 4, which is what the platform trial is all about, with fewer people on placebo. So we were able to share that. We ruled out that 2 of them really didn't work, regimen A and B, and shouldn't move forward. And then we have 2 that actually should go forward. Both regimen C and D have positive phase 2 data, and both those companies are now working with us and others to think about how to do the next study. So again, we couldn't have learned any of that without the participants in that study. So we're moving forward now. We'll have regimen E results, probably in the summer, I think. And then we're really excited to add a regimen F and G. And then this spring we're gonna take a couple days and invite some outside experts in platform trials and really look at the lessons learned from the first 4, whether we want to keep doing, whether we want to change. We really think this idea of a platform approach rather than a single study at a time is the way to go in ALS, given just the huge pipeline of treatments. So anyway, so please join us Monday. We're eager to share the results and then get people's input and questions.
Excellent. See you on Monday. Thank you so much. Thank you.